Hello everyone, today I am honored to introduce the research titled Weizmannia coagulans XY2 mitigates copper neurotoxicity via gut-brain axis modulation of tryptophan metabolism and oxidative-inflammatory crosstalk. Highlights copper interferes with tryptophan metabolism and 5-HT levels by modulating intestinal flora. Intestinal barrier breakdown and inflammatory response trigger nerve damage under copper exposure. Coagulans XY2 alleviates copper-induced neurotoxicity by targeting a multidimensional tryptophan metabolism antioxidant defense gut-brain axis network. Results regulation of copper-induced cerebral metabolic disorders in mice by Weizmannia coagulans XY2. Copper exposure caused severe impairment of cerebral metabolism. Intervention with Weizmannia coagulans XY2 significantly reversed the copper-induced imbalance of specific lipid profiles. This metabolic reprogramming is associated with reduced oxidative stress and improved neurological function. Tryptophan was identified as a common differential metabolite in both the brain and serum metabolomes. Coagulans XY2 modulated tryptophan metabolism, repaired serotonin biosynthesis, suppressed the kynurenine pathway, activated the production of indole derivatives, and restored cerebral serotonin homeostasis. Notably, there was no significant difference in cerebral copper content among the mouse groups, suggesting that copper may induce neurotoxicity indirectly via the gut-brain axis rather than acting directly on brain tissue. Copper exposure increased the level of superoxide anions in the colon and decreased the activities of glutathione reductase and catalase. Intervention with Weizmannia coagulans XY2 reversed the copper-induced overactivation of the NRF2 pathway and inhibited the expression of pro-inflammatory mediators. In addition, Weizmannia coagulans XY2 significantly restored the copper-induced reduction in TPH1 and 5-HT levels, and restored intestinal barrier integrity by upregulating the expression of tight junction proteins. Weizmannia coagulans XY2 improved copper-induced abnormalities in indole chemotaxis, impaired learning and memory, intestinal barrier damage, and dysregulated 5-HT levels in C. elegans. In the SKN1 knockout strain, the ameliorative effects of Weizmannia coagulans XY2 were lost, indicating that the neuroprotective effect of this strain is dependent on the SKN1/NRF2 pathway. Conclusion: Weizmannia coagulans XY2 confers neuroprotection against copper toxicity through a gut-brain metabolic axis. It regulates TPH metabolism, enhances antioxidant defense, and maintains intestinal barrier integrity. Coagulans XY2 boosts serotonin production, inhibits the neurotoxic kynurenine pathway, and promotes neuroprotective indole production to improve neural function. It also activates the SKN1/NRF2 antioxidant pathway and preserves intestinal barrier function to prevent oxidative inflammatory damage. These findings provide experimental evidence for probiotics to reduce metal toxicity via the gut-brain axis, extending beyond established models of copper-induced mitochondrial dysfunction. This work delivers a safe, multi-targeted strategy against copper-driven neurodegeneration with significant translational potential.
iMeta, a journal to change the world.